Hello, everyone. We will talk about how protein is digested. For this section, I'll make it two separate lecture because it can be too long. And so I'll break down the section from the introduction to this, what happens in the stomach. And then I'll start with another subsection to talk about protein digestion, starting from the small intestine to the end. And then also let me share the screen with you. Okay. So working on it. Hope you can see the screen with a white background. Okay, great. Right, let's talk about the protein digestion, how protein is digested. Before we get there, I'd like to give you some overview and share the remind on the structure of the gastrointestinal tract, and then we'll move to how the steps of protein digestion. Here, let's look at this picture that I took already. Oh yeah, years ago. I took this photo in 2013. My grad students to help me to have this gastrointestinal tract dissected, separated. This is a GI tract from 6 weeks old pigs. And pigs here, I mean, the pig industry, we wean the pigs at 3 weeks of age, and then this is 3 weeks after that. So it's not that big, the small pigs. If you look at this, the tract, you see the stomach. Prior to the stomach, you see the very short section of the esophagus coming from the mouth. The esophagus is connecting the mouth to the stomach, and we spend some time to talk about the stomach here. But then the stomach is connected to the small intestine, very long small intestine, case of most of the animals, including human pigs and chickens, the long small intestine. But the beginning portion, it is called the duodenum, and this is where this is a section of the small intestine connected from the thyroid bag of the stomach, and also the pancreas. Pancreatic duct is secreting the secretion into the lumen of duodenum. So this is the very beginning part of the small intestine. About 80% of the small intestine, in the case of mammals, is called the jejunum, and this is where most of the food digestion occurs. And digest our food, we spend a long time in this lumen of jejunum. As it goes from the beginning to the end, it will spend time and react with digestive enzymes, and food and feed particles will be converted, digested to the nutrients, and this nutrient will be absorbed into the body in this jejunum. So most of meaningful digestion and the nutrient absorption will happen in this jejunum. In that way, the jejunum is a very important portion of this gastrointestinal tract in the case of mammal. And that's the reason I say mammal is so avian will have a different, little different classification, characterization of the small intestine. So we'll talk about that in the next one. But the terminal site of the small intestine of the mammals is called ileum, and this is very short. Mammals, it will be just a few inches or less than 10 centimeters of the last section of small intestine, connecting it to the cecum or appendix or the large intestine here. most likely in the G-ileum, there's no real active efficient nutrient absorption occur, but they are connecting the small intestine to the large intestine. In the case of animals, the first part of the small intestine, large intestine, the small intestine is connected to the large intestine. First portion is called the cecum. In the case of a human, the cecum it's not really functional, and we call it the appendix is very small. But in the animals, non-human animals, the cecum is actually, it could be have a function of storing the food, digest for a short time, and this digester will be fermented by microorganisms. But this is pouch in small temporary space, but it immediately connected to the main portion of the large intestine. It's called colon, and the colon is the place where the digester is after nutrient absorbed, the leftover from the small intestine is spend time here, and they are fermented, utilized by microbial enzymes. So you see the microbial fermentation occurs in the colon, and they have this microbial enzymes and microorganisms who will utilize fibers or undigested protein and starch and so on into their into the nutrient for their own bacterial use, but also they make metabolites that can be useful, that can be absorbed, utilized by the host animals. Okay? The end part of the large intestine is the rectum with a very thick blood wall, with a highly muscular blood wall, and connected to the anus and the undigested portion of all digested will be excreted as feces. So this is like a good picture of a gastrointestinal tract of monogastric mammal. Mammal that is monogastric in this case. And there's no woman here. Okay? If I move to the avian species, this is an example of gastrointestinal tract of 8 broiler chickens. Okay? I borrowed a photo from University of Kentucky Extension Service, and you can find this photo online as well. Very, very famous, popularly used. Okay? Sorry that I include the head of the chicken right here, but it's connected to the esophagus. In the case of avian, they have very unique structure called crop. This crop is enlarged portion of large portion of this esophagus. Most likely, the crop is located in the middle of the esophagus. Okay? So crop is not a stomach. It's a part of the esophagus that is enlarged and can store their ingested food temporarily, but also it's well shown that this food in the crop will have a chance to be fermented here there as well. Okay? It's not a rumen, it's not a stomach, but there's a crop in the middle of the esophagus. But after the esophagus is connected to the stomach of avian species, they have a two part of a stomach. I call that first part is called proventriculus, and which is before the ventriculus, proventriculus. And the second part will be called ventriculus, and also called gizzard. Gizzard, that's another term, more common term used. Okay? So proventriculus is the section where the enzymes are secreted and the acids are secreted, and the ventriculus is where the digestion and the secretions are mixed with the muscle contraction and movement. Okay? And right after the ventriculus, you see the section of the small intestine mainly attached to the pancreas. So we call this, it looks like the loop is connected each other. So we call that duodenal loop, and the section of small intestine surrounded, connected to the pancreas is called duodenum. And this is very look different from the mammals that I showed you previous slide. Okay? So it is rather easier to define, okay, where duodenum starts and ends in the case of avian, because I can identify the pancreas and the intestine surrounding the pancreas, which is the making a loop. So that is the portion of small intestine called duodenum. After that, they have a long small intestine until you see a pair of this blind-ended structure called ceca. So there are two, there's a pair, so we call it ceca, plural of the cecal, so ceca. Okay? So that's the beginning of large intestine. But if you go back and look at the small intestine, we already talked about duodenum. But if you look at the small intestine, remaining part of small intestine, in the middle of somewhere, we will see the very small button that you can see that is called Meckel's diverticulum. And it in the poultry nutrition here, we defined it that small intestine before the Meckel's diverticulum, we call it jejunum. But after the Meckel's diverticulum, we call it ileum. So the concept of jejunum and ileum in avian species is different from the mammals. Right? So in the avian, both in the jejunum and ileum and also duodenum, that's where the food will be digested and into nutrient, and nutrient will be absorbed, both including the ileum. Okay? So in ileum is a lot longer. There's a larger portion of the small intestine in the case of avian compared to the mammals. The mammals' ileum is very short, last end part of the small intestine. And so you see the difference, how we define the ileum between the avian and the mammals. So at the end of the ileum, you can see a pair of the structure called the ceca. Okay? And once the digesta entered this ceca, mostly it will be largely extensively fermented, and the remaining parts may be restrictedly come back to the large intestine. But this is where the fermentation, micro-fermentation occurs in the case of avian. They have a very short large intestine, okay, and that is connected to the end part of the GI tract. It is called cloaca, different from the anus or rectum, because in the case of avian species, the urinary tract and the large intestine is connected, and there's excreta are excreted together. And so we call that cloaca. So I hope you can compare the gastrointestinal tract of the mammals and avian and see the difference. Okay? This is the overview. This is the overview of how protein is digested. Okay? It's very conceptual. The numbers is very conceptual and doesn't apply to in details, but just in general purpose is okay. And this is from they are optional in the reading material here. Let's say an animal or human eat a food, so diets. Let's say there's 100 grams of protein is ingested, okay? And the middle part here, that's the lumen, the space in the gastrointestinal tract, and this is the small intestine, and there's rectangular things, looks like bricks, and that is enterocytes, and they both outside here, and both side is the inside of the body. So protein enters and come to small intestine, and the enzymes from pancreas will be secreted, and one of them is called the trypsinogen converted to trypsin. So this trypsin will be endopeptidase and will digest the protein into peptides, and other enzymes will further digest peptide into the single amino acids. And our body effectively absorb small peptide, like dipeptides and tripeptides, or single amino acid into the enterocytes. So this is done, and it's very effective. Protein is effectively digested to nutrients, single nutrient, and peptides and amino acid are absorbed into the body. And those enzymes, the trypsin as an example, there are other enzymes. They're like chymotrypsin or elastase. We'll talk about that later too. But these enzymes are believed to be also degraded or digested in the small intestine into the peptides and amino acid and being absorbed. However, if you see, you study hard, study deeper, and there's the concept. There's a concept called enteropancreatic circulation. I put the question mark here. It's indicating that possibly some of these digestive enzymes, including trypsin and other enzymes like amylase, will be reabsorbed through the small intestine, probably more likely in the ileum, and go back to the pancreas and then convert it back to the zymogen, which is trypsinogen, and being secreted here. So traditionally, we're thinking that those endogenous enzymes, secreted enzymes, will be degraded after it worked and into the amino acid. However, there are thought that some of the enzymes will be recycled. Okay? Yeah, I put the question mark. I encourage you to do your own research about this enteropancreatic circulation, and because there's some research going on here. So I put something called endogenous protein. That means some proteins are secreted from the body to the lumen of the small intestine. That is including enzymes of protein, also some dead cells from the villi. So all those are proteins contributed from the host animal. Typically, if the animals are ingesting 100 grams of protein, but 70 grams of endogenous protein will be released, making 170 grams of protein in the lumen of the GI tract, in the lumen of the small intestine. Out of this 170 grams, 100 here, 70 here, out of 160, 170, almost all, which is 160 grams, will be absorbed into the body as a form of amino acids. Okay. The remaining 10 grams are supposed to be undigested and excreted later as a part of feces. So 100 grams of ingestion and 10 grams of excretion. So you can simply say 90% of digestibility. However, there's a contribution of endogenous protein here, so it is a little more complicated than the simple 90% digestibility. Okay? If you absorbed the protein as an amino acid, that will enter the body. Then you go to the circulation, go to the liver, and also they'll be actively used to make a protein. Also, will be delivered to non-hepatic tissues like muscle and other tissues, and used for to make a protein. Okay? So this is the overview of what would happen there in terms of protein digestion. So when we say digestion of protein, this is the process of cleaving the specific bond between amino acid, because protein is a sequence of amino acid. So made of amino acids that are connected together to make protein. So this connection, the bond, is called the peptide bond. Okay? So protein is made of so many amino acids connected by peptide bonds. So protein digestion, it is the action of hydrolyzing the peptide bonds. And so then what is the peptide bond? Those are the covalent bond between the carbon and nitrogen. If the carbon is coming from the carboxyl group of one amino acid, the nitrogen is coming from the amine group of the next amino acids. So the carbon and nitrogen will make a covalent bond, and that is peptide bond. So the protein digestion is action of cleaving this peptide bond. Here, one thing that you learn from the biology or biochem or chemistry, when we draw a section of peptide made of amino acid, always I'll put the nitrogen on your left side and the carbon on your right side. This nitrogen and it's called N-terminal amino acid, N-terminal amino ends, and the carbon on the right side, that direction is called the C-terminal or carbon terminal carboxyl end, amino end, carboxyl end, or N-terminal or C-terminal. Okay? So always put the amino end on the left side, carboxyl end on the right side. Okay? That's the agreement. Right. So the action of cleaving the peptide bonds, depending on the types of enzymes, it can cleave the peptide bonds in the middle of a peptide or at the end of peptides, either carboxyl end or amino ends. So if an enzyme cleave a peptide bond in the middle of a structure of the protein here, in the middle of peptide, it is called grouped as an endopeptidase, digesting peptide from the inside, endopeptidase. If an enzyme is cleaving peptide bond at the amino end or from the carboxyl end, that is called grouped as an exopeptidase, enzyme digesting peptide bond from the outside, exopeptidase. Okay? There are a good number of endopeptidases when you talk about enzymes for protein digestion, including pepsin and trypsin, we'll talk about later. The case of exopeptidase, there's a good example called the carboxypeptidase, and we'll talk about that as well. Okay, let's talk about protein digestion in the mouth and the stomach, and then we stop and then move to the second section. In the mouth, in the animal, in the mouth, there's no protein digestion happening in the mouth. There's no enzymes involved, even though there will be physical breakdown of large chunk of food into smaller pieces, okay, by chewing, but no enzymatic digestion occurs. However, in the case of starch and lipids, some limited digestion of starch and lipid can happen in the mouth, but there's no protein digestion in the mouth. Okay? Now, this food will be ingested and move through the esophagus and be stored in the stomach. Okay? And when I say the stomach in the case of monogastric mammals, in the case of avian, they are monogastric. I'm talking about proventriculus. Okay? What happens in the proventriculus? In the case of the ruminants, I'm talking about what happens in the abomasum, which is the last the portion of the stomach, or I should say true stomach, in case of ruminants, excluding pregastric chambers. Okay? Okay, so once the animal ingests food, and there will be a distension of the stomach. Stomach will be full with the digested food, and these peptides or part of proteins in the stomach will stimulate the secretion of hormone, okay, called gastrin. So gastrin is the name of one of the digestive hormones controlling the digestive process. Okay. The gastrin is secreted from cells called G cells. Those G cells are found in the pyloric region, the end part of the stomach. I'll show you a photo soon. Okay. And also, these G cells are also found in the duodenum, which is the first part of the small intestine. And the secretion of gastrin from the pyloric region and duodenum of the small intestine will be inhibited when the digestive pH goes down. Okay. Under acidic condition, gastrin will not be secreted and will stop. Okay. So that is gastrin, which is the hormone. We'll come back with this. And in the stomach, in the middle part, the main portion of the stomach, we call the fundic region, okay, compared to pyloric region. And there's a fundic region. If you look at the surface of the fundic region, you see many pits or glands. We call it gastric pit, or in other terms, called the fundic glands. Okay? And there are, soon I'll show you a photo here, a picture, but there are different types of cells found in this gastric pit or fundic gland. One is called parietal cells, parietal cells. Another types of cells is called sheath cells. They have different functions. The parietal cells will secrete the gastric acid, hydrochloric acid, into the lumen of the stomach. In the case of sheath cell, they'll secrete some kind of enzyme, inactive enzyme called pepsinogen. The pepsinogen is secreted into the lumen of the stomach, and the pepsinogen will be converted to pepsin by gastric acid, hydrochloric acid. Right? So parietal cells and sheath cells actually are working together. They are found in the gastric pit or fundic gland in the fundic region of the stomach. Here, I'm showing you the structure of the stomach. In the case of a pig here, so human is very similar. Avians are different. In the case of abomasum, will be similar. Okay? And this is esophagus, and that's very small portion of the stomach connected from the esophagus called esophageal region. This is a very beginning portion of stomach. One interesting thing is there's no mucin secretion in this esophageal region. But then the first portion of stomach called cardiac region, the main portion is called the fundic region. And the last part is called the pyloric region. The pyloric region will be connected to the pyloric belt, controlling the movement of food digested from the stomach into the duodenum. Only except for the esophageal region, all others, cardiac, fundic, and pyloric regions, they all secrete the mucins. So the mucin will covering the surface of the stomach to protect some tissue from digestion. Okay, and at the end of pyloric region, through the pyloric valve, is connected to the small intestine, to the bladder. Right? So that's the review of the structure of the stomach. If I show you the close picture of the gastric pit or fundic glands found in the fundus region of the main part of the stomach, okay. You can find parietal cells, okay, or sheath cells. Okay. They parietal cells will secrete the hydrochloric acid into the space in the pit, and they'll be secreted into the lumen of the stomach. In the case of tube cell, they secrete the pepsinogen. We'll talk about this soon. Pepsinogen is secreted into the lumen of the stomach, okay? But it doesn't do anything yet. This is an endopeptidase, but it's not active yet. Okay? They should be activated. Okay? This activation is done by hydrochloric acid. The low pH will convert pepsinogen to pepsin, which is an active endopeptidase. Coming back, let's talk a little more about gastrin, which is a hormone, and what it does. Gastrin, okay, will be secreted when after animals eat and the food is full in the stomach. So the presence of peptides and amino acid in the stomach will cause the release of gastrin from the pyloric region, G cell, and also G cell in the duodenum. Okay, will be secreted from there. And gastrin will stimulate. What it does, the gastrin will actually stimulate the secretion from the gastric pits. So hydrochloric acid and pepsinogen secretion will be controlled, stimulated by gastrin. Also, gastrin will encourage the proliferation of gastric epithelium, repairing the surface of the stomach by making new cells. Okay, once gastrin causes the secretion of hydrochloric acid from parietal cells and pepsinogen from the tube cell, okay, and this is what happens after that. The hydrochloric acid from parietal cells will reduce the pH of the digesta in the stomach to around 2 to 4, very acidic. Okay. What it does, this hydrochloric acid will have a function to sterilize, to kill the bacteria coming from the food. Okay. Another important thing is denaturing the protein, because a low pH will cleave hydrogen bonds within a structure of the protein. So protein is in the three-dimensional structure. Most likely, under low pH, this protein will become very long, linear polypeptides. Okay? So it's causing the unfolding of protein chains. Once protein becomes polypeptides, now the enzyme can cleave the peptide bonds effectively. Okay? So it increased enzyme accessibility. Now, pepsin, coming from chip cell, is secreted as a pepsinogen. Okay? It is an inactive form of enzyme. So when enzyme is secreted as an inactive form, we call them all together zymogen. This zymogen is an inactive enzyme. So this inactive enzyme needs to be activated. And how does it happen? Is by the hydrochloric acid. Under low pH, this pepsinogen will be converted to pepsin. Need a low pH. Okay. This pepsin now is an active enzyme, is an endopeptidase. There are different types of proteases. Protease means enzyme digesting protein, proteases. This is a group of aspartic proteases. Okay. We'll talk about these different types of proteases in the next section. What it does, the pepsin, it will cleave peptide bonds. Randomly, what actually is not random, it will find aromatic amino acid. It will find hydrophobic amino acids, okay, in the middle of protein, in the middle of polypeptides, and will cleave peptide bonds within that. More specifically, it will hydrolyze carboxyl side of peptide bonds next to aromatic amino acid and branching amino acid in the middle of protein. So it will cleave cut very long polypeptide into shorter polypeptide, but still they are still big. Okay? So production, the end product will be large polypeptides. And what it does, it breaks down about 15 to 50% of all peptide bonds. So pepsin is very important, but it doesn't do complete digestion of protein at all. Right? So it will just cause the initial digestion. Okay, so I should talk just a little more. There's another protease called the renin. Okay. Renin is different types of enzyme with similar role to pepsin, but it is mainly secreted from the young suckling animal. Okay. And this renin will cleave the peptide bonds in the middle of the protein when they see the hydrophobic amino acid, but they will work under weak acid condition. Whereas the pepsins need very low pH, like 2 to 4, but renin can still function a little higher, but still acidic condition, like pH 5 to 6, okay, 4 to 6. Okay. On the right side, I'm showing you the structure of pepsin. Okay, this is actually pretty big protein made of 325 amino acid, 35 kilodalton, and this is the active site. This is where peptide bond will be cleaved. But in the case of pepsinogen, which is a zymogen, inactive form of pepsin, it is a little larger than pepsin because this active site is covered with another part of protein. So pepsinogen is surely bigger than pepsin, right? Okay, so we talked about the overview, and we talked about peptide bonds, and we talked about what happens in the mouth and stomach in terms of protein digestion. So we'll have a break a little bit, and then I'll continue protein digestion happening from the small intestine all the way now. Okay, thank you. I'll stop sharing. Okay, I'll see you soon again.